let's talk about DNA fingerprinting. So what is DNA fingerprinting? So coming to the DNA fingerprinting, that shows genetic makeup of the living organisms. Mainly it shows genetic makeup. Okay, so usually finding every human genome is very daunting and also very expensive. So for one human genome project, it is approximately costed around 9 billion of US dollars. And that too, many countries and 3 organizations came into play, that is US Department of Energy, National Institute of Health, Wellcome Trust by UK, and also by Japan, France, Germany, and China. So these are the countries that they have contributed, and others contributed for human genome projects. So that's why, so finding the human genome for each and every human being is very tough. So that's why, so we are finding the difference in the satellite DNA. So we have a DNA here, we call it as satellite DNA. So coming to the satellite DNA, so satellite DNA also we have two things, microsatellites and mini satellites. So coming to the satellites, so these are none other than the stretches of repetitive DNA which do not code for any specific protein. For example, if it is ATAC, so repeatedly you will be having same ATAC and again ATAC and again ATAC. So like this it would be repeated. So this will not code for any specific protein. So these non-coding sequences are the major chunk for DNA profile of humans, because this would be transmittable, means this would be heritable, and this depict a high level of polymorphism. You can see in this we have a polymorphism. So that is the basis of DNA fingerprinting. The meaning of polymorphism is you will be having same kind of sequence, but only one nucleotide will be different for each and every sequence. So that we call it as polymorphism. This is the main basis, and we have the high level of polymorphism in all kinds of tissues. As a result, they prove that is very useful in two cases. One is forensic studies. At crime scene, paternal disputes, any piece of DNA found at the crime scene can be analyzed of a level of polymorphism. So in non-coding repetitive sequence, after the DNA profile is traced, it becomes easier to find out the criminal scene. So by performing the DNA fingerprinting, for example, you can take hair follicles, skin, blood, sperm. So these things can be collected as a samples. If these samples are available at the crime scene, then it is very easy to perform the DNA fingerprinting. Apart from crime scene, we can also find the paternal unclaimed baby and conducting the paternity test also. So let's see the DNA fingerprinting steps. So what happens in this DNA fingerprinting? This was developed by Alec Jeffries. Alec Jeffries developed this technique, used satellite DNA. And he called that satellite DNA as VNTRS, variable number tandem repeats. So, and also as a probe, because it is showed high level of polymorphism. Because this variable number of tandem repeats has showed high level of polymorphism. And what are the steps that is involved is, very first thing, we have to isolate the DNA. After isolating the DNA, digesting the DNA, with restriction endonucleases. So restriction endonucleases, you can otherwise call it as molecular scissors. Right? And after separating DNA fragments, so how we are separating the DNA fragments is by gel electrophoresis. So we are taking the agarose gel in which we are applying the electric current. So that's how the DNA fragments will be separated. Now the separated DNA fragments will be taken as a print by blotting technique. So what is this blotting technique is separating the DNA fragments on synthetic membrane. On synthetic membrane, which is nylon. And hybridizing these fragments by using VNTR probes. Hybridizing the fragments using probes. Analyzing the hybridizing using autoradiography. So coming to the application, it is earlier the discussion, it is used mainly in the forensic test. And mainly in the paternity disputes. So, and also it also gives an idea of gene frequency or genetic drift. And mainly remember, in identical twins, DNA fingerprinting will be same. We can only able to find out them with normal fingerprints. Fingerprints are different, but DNA fingerprints is same for identical twins, right?